So let's talk a minute about what breathing is. So breathing is really just the mechanism that moves air into our lungs and pushes air back out of our lungs. That is breathing. Now, it's driven by pressure gradients within your lung, and these pressure gradients are driven by atmospheric pressure, that air pressure that is in the atmosphere versus that air pressure that is in our alveoli, which is called alveolar pressure, and the pressure within our pleural cavity, or what is called our intrapleural pressure. It is the atmospheric pressure that allows air to enter our lungs when we inhale and to leave when we exhale. And inhalation is also called inspiration, and exhalation is also called expiration. Some of you might have heard that when somebody passes away, you'll be told that they have expired. Well, that is actually referring to their last breath, their last expiration. Okay. Now, both of them, inspiration and expiration, are dependent upon the air pressure difference between that is in the atmosphere and that pressure that is in our lungs. Now, atmospheric pressure is the amount of force that is exerted by the gases that are in the air and that are surrounding the surface of, let's say, our human body here. Now, it is expressed in units that are called millimeters of mercury. And 1 atmosphere, or 1 ATM, is equivalent to 760 millimeters of mercury, okay, which is atmospheric pressure at sea level. So if pressure is lower than 760 millimeters of mercury, then that is what is called negative pressure. When it is greater than 760 millimeters of mercury, that is what is called positive pressure. If pressure is equal to atmospheric pressure, then we express that number as 0. All right, so let's talk about the relationships between the intrapulmonary and the intrapleural pressure. So the intrapulmonary is sometimes also called the intraalveolar. So that pressure that's in our alveoli versus the pressure that is in our lung tissue. So again, the intrapulmonary or the intraalveolar is referring to the pressure of the air that is within our alveoli. And the pressure is going to change throughout different phases of our breathing. Now, the trachea. Remember, when we breathe in, air goes in our mouth. It goes down our trachea into our bronchi, where it splits into the bronchi, and then it splits further into the bronchioles. And then the little air sacs at the end, those are the alveoli. Now, the trachea, the bronchi, and the bronchial tubes, they connect the alveoli, those air sacs, to the atmosphere, so that the intrapulmonary pressure or the intraalveolar pressure is always equalized with atmospheric pressure. Now, when we talk about the intrapleural pressure, this is the pressure in the air within our lung cavity or within our pleural cavity, and it's between our visceral and our parietal layers. Okay, so let me give you an example of the difference between the visceral and the parietal pleura so that you understand that these serous membranes are extremely important in protecting the lungs as well as lining the cavities of our lung. And then there's a serous fluid that's between the parietal and the visceral pleura so that it is helping to cushion, and it's also helping to keep down friction, because what happens when you have a lot of friction? When you have friction, you are producing heat. So the visceral and the parietal pleura with the serous fluid between it is going to help to decrease friction. It's going to decrease heat. It's going to help to protect our lungs from any kind of impact. So let's think about this a second. When my fingers go in, all right, my fingers are the visceral. In this case, they are the lungs, but it could be our stomach. It could be our heart, okay? And on the outside here, where my pinky is at, that would be The parietal pleura, the membrane that directly is around my fingers, that would be the visceral pleura, because we're talking about my pleura or my lungs. And then the air that's in my balloon, if this was the membranes that were surrounding our lungs, that would actually be the serous fluid instead of the air that's in my balloon. All right. And think about it. This is all one membrane. So it's wrapped around and folding around my lungs. And again, it's going to be filled with fluid. So if I bump up against something, it's going to be protected. All right. So that is my visceral and my parietal. My visceral pleura surrounds my lung. My parietal pleura, it is surrounding my cavity. And between the two membranes is the serous fluid. All right. So anyway, similar to the intraalveolar pressure, the intrapleural pressure also changes during different phases of our breathing. There are certain characteristics in our lungs, like the intrapleural pressure is always lower than or negative to our intrapulmonary pressure, and therefore it's always lower than atmospheric pressure. Although the intrapleural fluctuates during inspiration and expiration, the intrapleural pressure remains approximately negative 4 millimeters of mercury throughout your entire breathing cycle. Okay, so basically what we're saying then is that the pressure that's in your alveoli, all right, is going to always be lower than atmospheric pressure. And the pressure that is in your lung cavity, your alveoli sit in your lung cavity, the pressure inside your lung cavity, or the intrapleural, is the pressure that is going to remain approximately the same, or minus 4 millimeters of mercury throughout any time in the breathing cycle. Okay. So there are several forces that are going to be competing within the thorax that's going to result in this negative intrapleural pressure. So our lungs have this elasticity that creates one of the forces as the elastic pulls our lungs away from the thoracic wall. So our thoracic wall is here, and our lungs are going to be pulling away from that thoracic wall. Now, we also have surface tension of the water. Now, remember, surface tension, that's water molecules sticking to other water molecules through hydrogen bonds, right? So we have this surface tension of the water that's making up the alveolar fluid, which also creates this inward pull on lung tissue. Now, again, that's related to hydrogen bonds. Now, remember, our lungs, they produce this surfactant. It's produced by these alveolar type 2 cells, and the job of this surfactant is to help to break those hydrogen bonds so that the surface tension isn't so strong. Now, in our lungs, the water molecules are sticking to each other through these hydrogen bonds, and they are pulling in on the lungs. Now, if we don't break some of those hydrogen bonds, we're never going to be able to inflate our lungs. They are going to collapse, all right? So, and this is really important in terms of COVID-19, for example. In fact, what happens in COVID-19 is the SARS-CoV virus binds to the receptors on these alveolar type 2 cells. And when they do that, they enter into those alveolar cells and they take them over and they produce more of the SARS-CoV-2 virus. And then the cells undergo apoptosis and they burst open and they release that virus into your lung tissue where you can cough it out, sneeze it out, and so on. Now, the problem is that at the same time that it released the virus and the cell basically, imploded there, now you don't have the cells that produce the surfactant, which is why we are seeing people with pneumonia. Because anytime you have that buildup of water or other molecules like the SARS-CoV virus, the CoV-2 virus, I should say, then what we have is we have this buildup of stuff that's too much and it causes pneumonia. So that inward tension from the lungs is going to be countered by opposing forces of the pleural fluid and of the thoracic wall. And there's this surface tension on your lung cavity that is going to be working to pull your lungs outward. So you remember homeostasis, right? So we've got homeostatic mechanisms that are going to be required here, right? So if there's too little fluid in the pleural fluid, this would interfere with the creation of this negative intrapleural pressure. And we need that, right? So stop and think a second. A negative intrapleural pressure with positive atmospheric pressure is creating a gradient, correct? And that is what's going to allow us to inspire. So fluid levels are going to be carefully monitored. They're going to be drained or filled as needed by your lymphatic system. And so we have this outward pull of the chest wall opposing this inward pull on our lungs. Now, the outward pull is slightly more than this inward pull. And this is what creates that -4 millimeters of mercury intrapleural pressure, or that lung cavity pressure, right? Relative to the pressure that is in your alveoli. Now, or what's your alveolar pressure or your intrapulmonary pressure. So the difference between the two is that pressure gradient. And it actually has a name. It's called transpulmonary pressure. And it's what's going to help determine the size of your lungs. The higher that value is, the larger your lungs are. So I keep bringing up pressure gradients, and I'm bringing them up again. Inspiration or inhalation and expiration or exhalation are dependent upon differences in pressure between the atmospheric pressure and the pressure that's in your lungs. Now, there's a law here called Boyles' law. And what it does is it describes this relationship between the volume and the pressure of the gas at a constant temperature. And what Boyle discovered was that the pressure of gas is inversely proportional to its volume. So if a volume increases, then pressure decreases. If the volume decreases, then pressure increases. So pressure and volume are inversely proportional to each other. So again, if volume increases, then the opposite happens with pressure. Pressure decreases. If volume decreases, then pressure increases.